Good afternoon, everyone. My name is Basmah. Thank you for the nice introduction, and I would like to thank the conference for giving me this chance to talk today. And just for you to know, this is my very first talk in an international conference. So today I'm talking about hydroxybenzoic acid isomers and cardiovascular system. Okay, so I'm starting to with showing you a map of the worldwide death rate due to cardiovascular diseases. So you can see the really huge number in America, Middle East, Africa, and worldwide. So the increasing number of deaths due to cardiovascular system is not a small issue. So compare this graph with, okay, so this graph will compare the reason of death. Look at the yellow, the light. Is there a pointer? So, okay, look at the light, this one, the light blue and the light green. This one is showing you the rate of death due to cardiovascular disease, and this one is showing you the rate of death due to cancer. So comparing the people who are dying from cancer to people who are dying from cardiovascular disease, the number is way more in people who are dying from cardiovascular disease. So from this point, also, the American Heart Association is showing you comparing between cancer patient and a CVS problem. So the cardiovascular problem are apparently more problematic in worldwide condition. So why do we have this increased cardiovascular problem? So cardiovascular problem can be due to different types of diseases, either to be ischemic problem, which is reduced blood vessel, or to be a proliferative myocyte problem, or it can be inflammatory disease, it can be cancerous developing in the myocyte. So different types of problems that can happen inside the myocyte. And it will lead to the destruction of the cardiac muscle. So, from the American Heart Association, every 25 seconds of American people, every 25 seconds, imagine that a patient of American citizen will have a coronary event. Every 39 seconds, a patient will die because of a cardiovascular problem. And because of those tremendous number, it's really a huge matter we need to solve. And with all those risks, 99%, not by me, by the American Heart Association, are saying that 99% of the American population need to develop and take care of their heart health. So I'm kind of here today to give a small tips on how to develop the cardiac health of a patient. So we studied with my doctor, Dr. Bernard, Dr. Paul Ganguly. We did a paper on hydroxybenzoic acid isomers and their effect on cardiovascular disease. Starting the presentation, there's a term called nutrigenomics, which means what is the science behind the dietary component that we consume and their effect on our genome and our gene expression. Since the 18th century, there have been discovered of salicylic acid and salsine. So we all know it's the drug that has been widely used, like aspirin and those stuff. So those drugs were obtained from the willow bark extract. And the hydroxybenzoic acid are actually one of the derivatives of the salicylic acid. So the molecules that we studied is 2,3 hydroxybenzoic acid, 2,5, 2,4, 2,6, and 3,4 hydroxybenzoic acid. Those interesting properties, those molecules have an interesting properties in association with the ability to modify the cell signaling that introduces a multiple effect through a specific pathway, which is called the RNF2 pathway, which results in an enhancement of the endogenous antioxidant activity of the inside of the cell. Another molecule, which is also interesting to know, which is the sulforaphane, which is a very potent activator of NRF2 pathway and through the KEAP1 protein. So those also produces an antioxidant activity. This is the sulforaphane molecule, and you can see that it is a proteotype for inducing the activation of NRF2 signaling and block the chemical carcinogenesis. So, in the field of carcinogenesis, health reflects our ability to adapt to stress. And carcinogenesis field, there are different types of stress that a patient can be exposed to that can develop cancer or cardiovascular disease. Those different types of stress are metabolic, proteotoxic, mitotic, oxidative stress, and DNA damage. So RNF2 signaling pathway can be a target for preventing many of the disease. And how does it do this? A study has been done by Dr. Thomas Klenser in University of Pittsburgh, where they bring up a wild type of mice and they stimulate them by different types of molecule like altipaz and sulforaphane and different types of molecule, and they check the effect on the mice and how do they reduce the oxidative effect on the mice state. And also they do a knockout NRF2 signaling pathway to further study the effect of NRF2 pathway. And also they did tissue-specific, cell-specific KEAP1 knockout to study the amplification signal of NRF2 pathway. Okay, so how does this NRF2 pathway work, and does it actually have the ability to prevent us or help us to protect ourselves from the carcinogenesis, unavoidable carcinogenesis that we can be susceptible to through environment? Taking an example of sulforaphane and how does it work. Sulforaphane will target a specific molecule, which is cysteine 151, and it will, along with the KEAP1, they will block the CALT3. CALT3 they will inhibit the ubiquitination process, ligase, and then it will stop the proteasomal effect of RNF2, and this will lead to new NRF2 into the nucleus, and the new NRF2 into the nucleus, it will lead to the glutathione transferase production, and this glutathione will oxidize and will get rid of the pollutant that we can find in air or like benzene and carcinogenesis, and they will oxidize them, will conjugate them through glutathione, as the mercaptopuric acid will get rid of them. And the specific pathway of this NRF2, we will have the NRF2 activator over here. It will bind to a specific receptor on the cell. It will activate the kinase. The kinase will phosphorylate the NRF2, bind to KEAP1, and the phosphorylated NRF2 will bind to an area on our genome, which is the antioxidant response element. And this area, once it activates either through the phosphorylation or through another pathway, which is the oxidation of the thiol group on KEAP1, it will lead to the activation of the antioxidant, anti-inflammatory, and anti-fibrotic agents. All of them will be expressed through this area. So this is the gene that you just saw, that I just showed you in the previous one, this one. So this is the antioxidant response element. Link gene, once this area get activated, you can have protection against oxidative damage, against metabolic disease, against lipogenic stress, against damaged protein, and against glycosylation. So through those different types of pathway and signaling molecules. So after this brief introduction about NRF2, you can see that NRF2 protects against different types of disease through reducing the oxidative damage that can be caused by different types of oxidative stress. So it can protect against carcinogenesis, cardiovascular disease, strokes, ischemia, neurodegenerative diseases. A study have done previously, and I'd like to present this study because they've used a molecule, an activator of NRF2 molecule. They have an activator of this like really novel activator, which is the DH404. This activator will show you in the cardiac myocytes the effect of NRF2 activation. They did Western blotting and the effect in the nuclear and in cytoplasm, and you can see the difference between the cardiac myocytes, and the oxidative stress is potentially reduced. So we did a summary of, we studied each molecule of those hydroxybenzoic acids and what is their effect on the cardiac myocytes. There's different resources for each compound, and they're present in different types of food in different concentration. They have different mechanism of action, and definitely all of them will induce the activation of NRF2 pathway. And interestingly, the last two molecules, we found that they have the potential to activate, to act on HC1 and HCA2, which will inhibit lipolysis in adipocytes, which will control not only the problem in your hypertension or oxidative stress, but also problem with your lipid profile and dyslipidemia. So this, we created a summary of different types of food that you can eat with their percentage that is having different concentration of hydroxybenzoic acid, and you can look it up whenever you want. So the concentration, okay. So we have a concern raised up by if we can consume a pharmacologically active compound found in our food. So is it dangerous for us to consume different types of molecule? Yes, it can be dangerous. Why is that? Because our major concern would be on the effect of phytochemicals, which are those molecules, the DHPA molecules. They can be express an activity which is on the drug metabolism phase 1 enzymes. For example, the cytochrome B450. For example, look at this molecule, which is also an activator of NRF2 pathway. It can be a competitive inhibitor for the cytochrome B3A4, which can interfere with your drug metabolism. So you have to be conscious whenever you prescribe a specific type of drug. You have to be cautious what is your patient eating, and it can interfere with the metabolism of their own food. Hydroxybenzoic acid you can intake it not only from food product but also from your gut. So some type of food, like this study has been done by Dr. Zan and some colleagues, where they did take cranberry juice and give it to the people, and they found that specific bacteria in the gut metabolize a substance into metabolize the phenolics into 2,3 hydroxybenzoic acid, and which can be a source of us, like to have another source for the antioxidant effect of hydroxybenzoic acids. We can conclude that if we design a diet containing specific NRF2 activator that can act on KEAP1 thiol oxidation, or NRF2 allowing the efficient amount of translocation of NRF2 molecule into the nucleus, that will result in a normal redox state in our cell, which will keep a consequence decrease our chance of developing cardiovascular disease or even cancer cells. So diet with an increase, as I told you, the last two molecules, the 3-mono hydroxybenzoic acid and the 5,3 dihydroxybenzoic acid, that also can affect our not only oxidative status but also our lipid profile. So don't forget that, as Hippocrates said, let food you be medicine and medicine be your food. And this is my flight. I came all the way from here till here, and then from here till here, just to tell you, please keep calm and eat your broccoli or whatever oxidative food that you can. So thank you. Questions?